So I'm here to tell you about the connectome. It's a concept that I believe will revolutionize neuroscience in much the same way that the genome has revolutionized biology. And I've written a whole book on this. It will be available in the UK starting in 2 weeks, so I urge you to look in your bookstore. But you have the luxury of hearing about it directly from me. So what is a connectome? Well, this is a little worm called C. elegans. Biologists love to study it. It's just 1 millimeter long, and it lives in the soil. And this is its connectome. So I don't know what this reminds you of, but I just got off a plane yesterday, so it reminds me of the back pages of in-flight magazines. Just replace every city by a neuron and every flight between cities by a connection between neurons. Now, this is just a worm connectome, but to imagine yours, imagine 100 billion cities and 10,000 flights coming out of every city. That's the extent of the complexity of the network of neurons inside your brain. Now, why should we care about this connectome? Well, one thing is important, which is that your connectome is unique. No other person has the same pattern of neural connections that you do. And one of the great hypotheses in neuroscience is that your memories are somehow encoded inside that unique pattern of connections. And maybe other aspects of you, your personality. Perhaps if you have a mental disorder, that might be reflected in the unique pattern of brain wiring. And so in the TED conference that I met Herb and many of other people here 2 years ago, I summarized that in a slogan. I asked people to repeat after me. I asked them to repeat, "I am more than my genes. I am my connectome." It's a hypothesis, really. It's not a slogan that you can just say and believe in, because this is science after all. But this slogan summarizes the idea that human uniqueness may be really rooted in the connectome. So what's happened since then? That was 2 years ago. Well, we've been engaged in really what is an important struggle, a technological struggle, which is how do we actually map out the connections between neurons? And that worm connectome, with just 300 neurons and 7,000 connections, took over a dozen years of effort by a team of scientists to map out. So how do we have any hope of succeeding for brains more like our own? I worked on, well, we have to take brain images and we have to analyze those images, but the amount of data required is so huge, the task seems so daunting, that while the brain may seem awe-inspiring to many of us, sometimes it fills me with a kind of despair. How could we succeed in such a difficult task? 2 years ago, I put my hope on machines, that our computers would become smart enough to analyze the data automatically. But that's happening, but it's still too much data for computers to handle all by themselves. And so I've come to the conclusion that we need more than computers. We need you. We need all of you. I would like to see people on a mass scale organizing together and helping us analyze this data to map out connectomes. This notion of citizen science, which is very captivating to me, you've probably heard of Galaxy Zoo or Foldit, can now be applicable to neuroscience. And I've tried to think of a way of summarizing that in a slogan. So I came up with, maybe some of you can help me if some of you are in marketing, but here's my slogan. The people connected will never be defeated. So can you humor me and just say this? All right, let's try it. I'm trying it out today. First time ever. All right? The people connected will never be defeated. So that's the message I want to bring to you today, that in science, as in so many other fields, to solve the really difficult problems that face us, we have to come together. We cannot succeed alone. So what is the cause that might motivate us? So I founded this nonprofit called Wired Differently, which is going to sponsor and support the citizen science effort. And I'll tell you about the projects that we're planning now. So there's the projects we're doing, and there's the projects that we're planning. And so let me tell you about our ambitions. So last year I met a woman who has a son named Charles. Charles has Phelan-McDermid syndrome. He's a beautiful boy, but one of the symptoms is autism, and this is a very difficult thing for his mother and family to deal with. This is a little map of part of Charles's genome. And if you look at this map, and you're a professional, you'll see that a gene called SHANK3 has been deleted from Charles's genome. And this appears to be the reason that he has autism and the other symptoms of Phelan-McDermid syndrome. So recently, something amazing has happened, which is that a colleague of mine at MIT has created a genetically engineered mouse. He has damaged the SHANK3 gene in mice. And interestingly, these mice have symptoms that are reminiscent of autism. So on the right side, you'll see that if you take one of these mutant mice and put it in a cage with a regular mouse, it stays on the other side. So these mice have antisocial kind of behavior. And on the left-hand side, you'll see that these mice groom themselves excessively and repetitively to the point that their skin gets sore. So they also have repetitive behaviors. Now, we can't say that this mouse has autism, because autism in the end is truly a human kind of condition. But it does have some symptoms that are reminiscent of the human condition. This is a map, or this is an image then, of one of these mice's brains. And you can see that SHANK3 is highly expressed in a brain structure called the striatum. And so the obvious question is, could this region of the mouse's brain be wired differently in Phelan-McDermid syndrome? You know, if the mouse has similar kinds of symptoms, perhaps the brain wiring of the mice is also similar, and by studying this mouse, we might learn something about human autism. How can we figure that out? And here is where we actually need people's help, because, well, I'm not sure if any of you have ever had to deal with this kind of problem, but this is an artificial kind of tangled wiring behind some electronic equipment. But just imagine that millions of miles of wire are contained inside your skull. And a casual look at something like this will never tell you whether somebody malicious went and exchanged two wires, or whether somebody made an error in wiring this up. You actually have to trace the paths of the wires. That's the kind of task we're faced with in studying the brain. So here's a little piece of mouse brain. This is not yet, we're planning on doing this with the SHANK3 mouse, but here's a different piece of a mouse brain. It's embedded in a really hard plastic resin. And this is a really high-tech deli slicer. You can see there's a blue tub of water, and the world's sharpest knife, a diamond knife, is on the edge of this blue tub. And the brain slides back and forth, shaving off very thin slices of brain, thousands of times thinner than a hair. They float onto the surface of the water and they spread out. And then there's a white tape conveyor belt which collects those slices automatically. You may see gray smudges on the surface of that white tape. Those are the brain slices. This marvelous invention came from Ken Hayworth. It was developed in the laboratory of Jeff Flickman at Harvard University. You can take this tape now, this plastic tape, and glue strips of it to a silicon wafer. And now we're zooming in by a factor of 100,000 times to the resolution that we can see the brain's wiring. Except to see that wiring, we need to see it in 3 dimensions. So we can take images of many slices of brain and stack them up and form a 3-dimensional image. And then, if we start at one of the faces of this cube and color in a cross-section of one of the branches of neurons, the wires of the brain, and keep on following those cross-sections all the way through this volume, we can actually trace the paths of the brain's wires. That's a dendrite. And in green, there's an axon, and they contact each other at 2 locations. Those are the things called synapses. You may have heard of them before, but now you actually see them. And to verify that a contact between 2 neurons is a synapse, we have to zoom in and look inside at the molecular machinery. inside the green neuron, you may see little circles. Those little circles are vesicles of neurotransmitter. And so when the green neuron wants to send a message to the red neuron, it spits out the contents of one of those vesicles, and the red neuron senses it. So I've just demonstrated to you that if you have really high-resolution images of brain tissue, it's possible to find the synapses and trace the paths of the wires, so it stands to reason that we should be able to find the wiring diagram of the brain, or the connectome. And the imaging speed has been getting better and better. So over the last year, through technological innovations like I showed you, my colleagues have made great strides in improving the speed at which brain tissue can be imaged at this resolution. Here's a marvelous device that I saw recently in a visit to Germany. It is an experimental one. It's 61 scanning electron microscopes packaged in one microscope. And it is going to generate 1 petabyte every 2 weeks. That's 1 billion photos in your digital album. It will be delivered. The first 2 models will be delivered in 2014 to my collaborators at Harvard and in Munich. So what is 1 petabyte of brain images? Well, 1 petabyte is enough for just 1 cubic millimeter. That contains 100,000 neurons and 1 billion connections. And let me emphasize that the MRI brain scans you see in the newspaper, they blur all that into 1 pixel. So it's a huge difference in resolution. And if somebody were to color in the wires, like you just saw, inside 1 cubic millimeter, it would take about 100,000 years of manual coloring to finish, with no coffee breaks or naps. Now, we and other labs have been working on the AI to speed up this process. And the factor of speed up we get is about 10 to 100 times. But that's still 1,000 person-years of work. And here is where I think we can try to get the world involved, because this has come up actually several times in today's talks. Does anyone know how much time the world spends every day on Angry Birds? I read it recently. It's 600 years per day. So 2 days of Angry Birds would be enough to map out every connection in 1 cubic millimeter of brain tissue. And if we could make a game that was 1% as fun as Angry Birds, we'd be done in a year. And so I've become interested in this possibility of gamifying neuroscience. And so I'd like to show you then the first efforts to do that. Here's the site. It's called iWire, not little I, but obviously play on words. It's called iWire because the data set that we have, which is from my collaborators in Germany, Winfried Denk and his collaborators, the data set comes from the mouse retina. So that's the sheet of neural tissue at the back of the eye. And at this website, you can learn about neuroscience, but you can also play a game in which you help us map out the connections inside the retina. There's 5 players currently online. And you can see right here a visualization of one of the neurons that's currently its wires are currently being traced out by the players. And I've clicked on one of the people on the leaderboard to see the task they've done. Each of the blue cubes there is a cube of roughly the size that you saw before in the videos. And so this person is number 3 on this week's leaderboard because clearly they've done a lot of work. Okay, so what do you do inside? You can think about these users then as little people climbing on this tree and extending its branches. So what actually do they do? So if you sign in as a new user, you come to a display that looks like this. And what you're seeing on the right here is one slice through a little cube of retina. And we can move up and down inside that cube. So on the left, you see a 3D visualization of that. And so we're moving the plane up and down. And in blue, you see where the artificial intelligence has already colored in part of a branch of a neuron. And your job is simply to guide the AI. Just click to make the AI color a little more. So the AI has stopped. But if we click right here, it keeps on going. And in the 3D visualization we see on the left, the branch of the neuron has extended, and we've traced a wire through this cube like this. And this is an instructional task. So when you first sign up, you're learning to be an amateur neuroanatomist. So an expert has already done this. If you click in the wrong place, it turns red. And you go through a series of 15 instructional tasks. It gets harder and harder to find the missing branch. And when you finish, you graduate, and you go on to tasks that no expert has done before, and you're helping to advance science. Simple as that. Okay, first empirical finding. Some people like this. We have not a huge number of people, but we have some people who are playing this game quite a bit, to the point that I would say they're kind of addicted. And so why is that? Well, I think first of all, this is kind of mesmerizing. Some people have compared it to a lava lamp or watching clouds. And everyone has enjoyed coloring at some point in your life. And I knew we had something when one night I asked my girlfriend a question, and she said, "Don't bother me, I'm finishing a neuron." In fact, for several weeks, she was locked in an epic battle with Jose on the leaderboard. Every night, she would do enough tasks to get to be number one, and she would collapse in exhaustion. But Jose lives in Spain. I live in Boston. So when she would wake up in the morning, Jose would be number one because he had been working for 6 hours before she woke up. And so they would do this constantly. And several weeks after we opened this site, we created the forum, and that's how I learned who Jose was. Jose posted. He was one of the first people to start posting. We had a very active discussion forum, and he said, "Hello, fellow iWirer." They call themselves iWirers. And he said, "I play with my 7-year-old daughter." It's like, wow, we learned this is a family activity. And then he said, "Well, and not only that, my daughter and I found this piece of a neuron, and it looks like an ice skater." I was stunned. Because, the game, they were playing a game that I had set up, but now they were doing something that I never expected. This is the creativity of a community. So, of course, somebody else picked up on that and said, "I'll give you the finger." Okay, and so we realized from this that we should have an area of the site which is a gallery where people post the pictures of the neurons, and they caption them, and they vote them up or down, and have a lot of fun. So that we've only, I think, realized a small fraction of the potential fun that could be realized here. And those of you, I'm sure there's many of you people out there who are game designers and look at this and think, wow, that's really amateurish. I could do a much better job. And indeed, I think you could. And my goal in being here is to ask for your help. We're just a lab. I'm not a game designer. I'm a neuroscientist. I think that we could really go much farther with, if we could harness some of the talent in this room, get them working on a really engaging kind of goal like this. Now, some of the kind of contributions are really more technical than these creative ones of the images. For example, I was complaining to my developers for a while that, the image loading time is really slow. I'm having trouble using this site and so on. They said, well, it's a problem with your internet connection. You've got long latency packets. You've got drop packets. It's not our problem. So one morning I saw this post. And of course, W. Schall recognizes the long load time problems. So he recognizes the problem. He diagnoses the problem, not making use of HTTP pipelining at all. And then he says, he gives the fix. Use the keep alive header. Now, I don't understand any of this. I looked at my programmers. They look kind of embarrassed. They changed 3 lines. Problem solved. Incredible. It was an incredible experience for me, and it convinced me that this is the right way to go. That we really need to get people involved because there's so many talented people out there who can help us. Okay, so this is my ambition. We haven't gotten very far so far, but my ambition is that we have the chance to break down the barriers between research, education, and entertainment. That we could imagine an online community that tries to discover the causes of autism, all the while learning about the brain and having a good time while they're doing it. And so this is where I believe that you can help me. People can play this game, of course, but here everyone is an expert in thinking digital. I'm sure that there's so many of you out here who can think of ways to enhance this and make this happen, make it a reality. And that's why I say the people connected will never be defeated. Now, if we're looking for the unknown, we're learning about what exists, and if we're all enjoying each other's company, I would call that a quest. And when I was a boy, I read about mythic quests. I read about the Holy Grail, Sir Galahad finding it. And I read about Ponce de Leon, searching for the Fountain of Youth, never finding it, but exploring an entirely new world. And I would argue that the incredible technological progress that we've heard about all morning has positioned us. It has created us as a species that is ready for the greatest adventure of all time, the ultimate brain quest. And I hope that you will join me. Thank you very much.
So let me just, first of all, just to back up to more to the beginning, if you will, in the sense that just to make sure that I understand, and perhaps others in the room might be curious as well, is that the kind of the basis of your work is this idea that effectively genes are not all they were cracked up to be, right? This is not the answer is not in the genes in and of themselves, and that the answer lies, if you will, in the connectome, in these areas between the various communication structures, if you will, within the brain.
Yes.
But the challenge in being able to actually effectively prove that and build these models is such that you need to kind of mobilize, if you will, mobilize the cognitive surplus, to use a Clay Shirky's term,
right.
to kind of help crack this problem in anything like within our lifetimes or in a reasonable amount of time. Is that kind of roughly sort of what it's all about? Okay, cool. And is the way to get engaged, is it to go to iWire and check it out? And
that's the simplest way is just to go to the site. But I think if you go to the site, this kind of crowd will see so many ways in which it could be improved.
Yeah.
So many things that could be developed.
Yeah.
And so just play with the site and come talk to me. And I'm sure you can find a way to get involved. And that's what we're looking for, really.
Fantastic. Okay. Thank you very much, Sebastian.
That's all right.
Cheers.